Wouldn't it be amazing to fix a faulty gene to treat a deadly disease? Today, we're going to tell you about the fastest, cheapest, easiest gene editing method, CRISPR, which allows us to do just that. So, what is CRISPR? CRISPR stands for Clustered Regularly Interspaced Short Palindromic Repeats. Scientists found these short repeated segments of DNA in a bacterial plasmid, which were part of the bacteria's response against viruses. Now, scientists have hacked the bacterial CRISPR response to edit genes. Think of DNA as one long document. A gene is a sentence, and each letter is a nitrogenous base. For CRISPR to work, it needs to find and replace a gene or sentence, like we do while editing a document. First, we need to identify a familiar 3-base sequence near the gene we want to edit, like a road marker. This is the protospacer adjacent motif, or PAM sequence. In our document, it would be a common word that pops up a lot, like the. And the DNA sequence that we want to target to edit can be the sentence, "It was raining on the bluebell." In order to find and edit that target DNA near the PAM sequence, CRISPR uses gRNA, or guide RNA, edited by scientists to help guide the Cas protein, which acts as a molecular scissors, toward the specific target to make a double-stranded break. Think of gRNA as a Control F, or find mechanism, that looks for the target sequence next to the PAM. The Cas protein acts as a Control X cutting mechanism, or a way to add a space between the letters to completely change the sentence. For example, it can split the word bluebell into blue and bell, changing the meaning. It can even delete a whole segment of it, making it just blue. After this break in the DNA, it tries to repair itself, but this process is error-prone, resulting in the gene turning off its expression. However, adding a template DNA sequence that you want to insert in the gene through homology-directed repair can change the gene and the protein it creates. This is similar to the Control V, or pasting mechanism, or the find and replace mechanism. Then the word can become blue cracked bell, with cracked being the template DNA. This changes the meaning again and allows the altered gene, which will create a different protein, to be expressed. So, for a genetic disease like sickle cell anemia, CRISPR has the potential to edit a gene to mitigate the disease's effects or even prevent it. It can also be used by scientists to knock out genes to help understand their functions, aiding in further genetic research. Thus, this technology has immense potential to revolutionize science, medicine, and our way of life.